Hello, everyone. So, given the background we now have on mitosis, let's try to compare and contrast some of the similarities and differences between mitosis and a second mechanism that cells use to segregate replicated genetic information. So, again, as what we said earlier with mitosis, there's a specific goal in mind. The goal of a mitotic cell division is from an original cell to produce two daughter cells that are genetically identical to each other as well as to the original cell. So to maintain genetic constancy through the cell division. So that's not true for meiosis. Meiosis, actually, the goal is to produce genetically diverse cells at the completion of the process. And there are, different ways that that particular goal is achieved, as we'll talk about some of those details. So again, just to put everything in the context of this depiction of the life cycle of a human, as we've discussed previously. So again, we all start out as a single cell. You're able to regenerate an individual subsequent to a fertilization event through mitotic cell division. So to create many numbers of cells, trillions of cells, actually, all of which assemble the new individual, the body of the individual, and all of those cells, those trillions of cells, contain the exact same genetic information as that original zygote. So again, in adult individuals that can reproduce, have the ability to reproduce, and specifically for humans, sexual reproduction, within the gonads, there are specialized cell types that have the ability to proceed through a meiotic event to produce on the male side either a sperm and on the female side an ovum or an egg. So these two reproductive cells, again, have to be generated through a different mechanism than mitosis. Again, what we have to do is, because there's two parents involved in producing through the fertilization event the offspring, is that there's a need for those reproductive cells to contain half the genetic information of all of the other cells of the two parents themselves. So again, what meiosis does is minimally what's required is that you have to have a diploid cell. And then at the completion of meiosis, you generate haploid cells. And even more specifically, you create a population of haploid cells that are genetically diverse, genetically distinct from each other. So again, we're going to talk about some of these specific details that allow for this particular goal or goals to be achieved through the process of meiosis. So again, just to put meiosis in the context of the cell cycle, just like we did with mitosis, the cells that commit to a meiotic event, these specialized cells within the gonads, spermatogonial cells on the male side and oogonial cells on the female side, those are diploid cells. Those are 2N cells that, again, can proceed through meiotic events and maintain constancy of genetic information through those mitotic cell divisions. But eventually, these specialized cell types receive signals that allow them to commit to a meiotic event. So again, the cells prior to the meiotic cell division are an interphase, just like we've described previously. There's a G1. Once the commitment to a meiotic event has occurred, you'll transition through an S phase, when all of the genetic information within the nucleus of the cell is duplicated or replicated. You then transition through a G2, and then you enter into a meiotic event. So meiosis, one of the fundamental distinctions between mitosis and meiosis is that in order for meiosis to go to completion, it actually requires two cell divisions. So with mitosis, we started with a single cell. When the process was complete, you ended up with two daughter cells. For meiosis, because there's a requirement for two cell divisions, you start with an original cell, an original 2N or diploid. And after those two cell divisions, the final haploid meiotic products those haploid meiotic products is a total of 4 cells. Okay, so in describing this process of meiosis, that means that there's a meiosis 1, the first cell division, meiosis 2, the second cell division. And in describing the details of these 2 cell divisions, you use the exact same descriptive mechanism as you do with mitosis. So in other words, each of those cell divisions will consist of a prophase, a metaphase, an anaphase, and a telophase. And the characteristics of all of those stages for a meiotic event, whether it be meiosis 1 or meiosis 2, the general characteristics of those 4 different stages are exactly the same as what I described for mitosis. So I want to point out here is that once the cell has made the commitment to divide meiotically, the very first early stages of meiosis is going to be prophase 1, prophase of that first meiotic cell division. So there are very important events from a genetics perspective or a genetics viewpoint that are occurring during prophase 1 of this first meiotic cell division. And so actually cell biologists or cytogeneticists have subdivided prophase 1 into 5 different substages. And that's what's being represented here. So there's leptonema, zygonema, pachynemia, diplonema, and diakinesis. Okay, so going from leptonema to diakinesis is simply transitioning through prophase, the first meiotic prophase, prophase 1. And so again, going from leptonema to diakinesis, what happens is that the nuclear envelope dissolves, the chromosomes condense from a chromatin-like state to, very readily visible structures. Individual chromosomes are recognizable. And then lastly, there's a spindle apparatus that is assembled. The chromosomes, the condensed chromosomes, become physically attached to that spindle apparatus, and they are eventually pushed and pulled until they align at the central axis of the spindle apparatus. And when that is completed, you then transition into metaphase. So we'll get back to, again, the details of this process, but again, just describing meiosis generally, this is what happens during prophase 1. Again, the same characteristics as I described for meiosis. Once those events are completed, you are in metaphase 1 of meiosis. Then you again, the same characteristics as I said for mitosis. Those chromosomes are now aligned at that central axis of the spindle apparatus. Then you transition into an anaphase where you get segregation of replicated chromosomes. And then you have telophase. And again, telophase, what happens during the telophase is that you reform a nuclear envelope around the chromosomes that have aggregated at the 2 poles of the spindle. The chromosomes decondense into a chromatin-like state. And then the spindle apparatus is no longer needed, so it's broken down and dissolved. Just like with mitosis, during telophase, there is a simultaneous cytokinesis taking place that segregates the cytoplasmic components of the cell. The end result is that you have 2 daughter cells from that first meiotic cell division. So those 2 cells will then enter into the second cell division of meiosis, or meiosis 2. And again, they transition through a prophase, the same characteristics as I've been describing. They transition through a metaphase, they transition through an anaphase, and they transition through a telophase, and then subsequent or simultaneous cytokinesis. So the end result of these 2 cell divisions, again, starting with a single diploid cell, at the completion of meiosis is you produce 4 cells. All 4 of these cells are now haploid, and we'll see how that comes about in a few minutes. They're all haploid and they're all genetically diverse. They're all genetically distinct from each other. Okay, so what I want to do now is, now that I've described the overall process, all of the different stages of a meiotic event, I want to look at each of these stages individually and talk about them in a little bit more detail. So again, for our example, we're going to start with a 2N cell, a diploid cell with a total of 4 chromosomes. So again, what that means is that there's 2 pairs of homologous chromosomes within this hypothetical organism's genome. And what we have depicted here is a cell, that diploid cell in interphase or at interphase. So again, when you look in the nucleus of the cell, you can't distinguish individual chromosomes. So you have this chromatin-like material. So again, clearly interphase, unable to distinguish between G1, S, or G2. But now we're going to assume that this particular cell, whether it be a spermatogonial cell or oogonial cell for a human, has committed to a meiotic event, and it's now going to enter into prophase 1 of meiosis. So again, remember, prophase 1 is divided into 5 different substages. So what's happening during the very first substage? This is again leptonema. Leptonema means thin threads. So not much has really happened. This is very early prophase 1. So all that you can distinguish, as being different from interphase is that the chromosomes are now becoming visible because they're beginning to condense. So it no longer looks like sort of the generic chromatin, but again, you can begin to recognize individual chromosomes. So that's really the only distinguishing characteristic of leptonema. So then you transition a little bit further into the process of prophase. So you then enter into the second stage or substage that we call zygonema. And this is very important here. There's a very important event taking place that we didn't see with mitosis, during a prophase in mitosis. And what's happening during zygonema is that homologous chromosomes are pairing up with each other. It's just at this point, it's, again, it's early. It's a very loose association or a rough pairing of homologous chromosomes, but they're clearly interacting with each other. And again, remember, in mitosis, at any of the stages of a mitotic event, is that there was never any association between any of the chromosomes, whether they be homologous or non-homologous. All chromosomes proceed through mitosis totally independently of every other chromosome. So that's not true in meiosis. You can actually see very early on, this zygonema stage of prophase 1 of meiosis is that homologous chromosomes pair up with each other. So these paired structures are at this point are what we call bivalents, a two-membered structure. Okay. So then everything else, that occurs during prophase is taking place as well. The nuclear envelope is continuing to break down. You can see now out in the cytoplasm, the centriole has duplicated. The 2 poles are migrating to opposite sides of the cell, and the spindle apparatus is now in the process of being assembled. Okay, so from zygonema, you then transition into pachynemia. And what happens, that distinguishes pachynemia from zygonema is that the chromosomes have further condensed. So you can see a little bit more detail about their makeup. And if you remember, all of these chromosomes replicated themselves during the S phase of the cell cycle. So it's during pachynemia that you can first recognize the replicated nature of these chromosomes. And again, remember that replicated nature of the chromosomes is such that now each replicated chromosome consists of 2 sister chromatids. I described that, that's true for mitosis as well. So now in these original bivalent structures, you can now see the replicated nature of the chromosomes. And so those paired structures, those paired replicated homologous chromosomes consist of 4 chromatids. And so you now call the bivalent a tetrad. In this case, our example 2n of 4, there's going to be two tetrads that are forming during, or that would be forming and recognizable during pachynemia. So the association between these two replicated homologous chromosomes, remember the initial bivalent formation was just loose pairing, a loose association. Now they're becoming more tightly associated. So we call this a tight synapsis. Okay, so then, so that's the important event that distinguishes pachynemia. Okay, so then everything else, as far as prophase, the nuclear envelopes breaking down, spindle apparatus is becoming more fully assembled. So again, just to introduce a little bit of terminology again, here's a schematic of a sort of a generic tetrad. So two homologous chromosomes, they're replicated. They're now paired up with each other. So again, the tetrad structure consists of 4 chromatids. And again, remember, each chromosome consists of 2 sister chromatids. So these 2 chromatids of this purple chromosome, they're sisters, they're genetically identical to each other. The 2 green ones, again, are genetically identical to each other. They're sister chromatids as well. However, if you were to compare a green chromatid with a purple chromatid, they would be genetically similar to each other, but not necessarily identical, because of allelic variation that can exist on those 2 different homologous chromosomes. So what that means is that if you were to compare, say, a purple chromatid to a green, you would describe them as being non-sister chromatids because they're genetically similar, but not necessarily genetically identical to each other. Okay, so that's pachynemia. So then again, you progress further into prophase 1. You're now in diplonemia, the 4th substage of prophase 1 of meiosis. And the distinguishing characteristic here is now within these tetrad structures that have formed, is that you can see points of overlap between non-sister chromatids. And so these points of overlap are individually are referred to as chiasma, and collectively are referred to as chiasmata. So again, those are very important. The formation of those chiasmata, very important from a genetics perspective. And the reason for that, again, remember what we said was that one of the goals of meiosis is to produce genetically diverse products, final meiotic products. So what happens within these chiasmata is that there's actual physical exchange between non-sister chromatids. So this is a process that we call genetic recombination. And again, we'll talk more about that in later lectures. But what you need to be aware of now is that genetic recombination events take place within the tetrads that form during prophase 1 of meiosis. And it allows for achieving, one of the fundamental goals of meiosis, which is to create genetically diverse haploid cells as the final product. So the genetic diversity is produced through these recombination events. So I should say as well is that these chiasmata are first visible during diplonemia, but actually those recombination events are taking place in the previous stage, pachynemia. You just can't see them yet because the chromosomes have not condensed enough. So it's the same thing, a bivalent is a tetrad, but because the condensation of the chromosomes is not to the extent that you can actually recognize the tetrad structure. So it's the same sort of idea. So then in diplonemia as well, you can see the nuclear envelope is continuing to break down. The spindle is more fully assembled. Again, these tetrad structures at this stage would, again, physically connect to the spindle apparatus through their centromeric regions. And then they would start to be pushed and pulled until they're aligned at that metaphase plate when the spindle is fully assembled. So the last substage, this is now late, the end stage of prophase 1 of meiosis is what we call diakinesis. And, again, you can see at this point now the spindle is fully assembled. The tetrads are migrating towards that central axis of the spindle. The nuclear envelope is gone. The chromosomes are now fully condensed. And really the distinguishing characteristic in diakinesis is that those points of crossing over, of recombination, the chiasmata, actually what they do is once they form, recombination takes place, and then the chiasmata migrate towards the free ends of the chromosome arms. So it's the movement of those chiasmata. It's called a process called terminalization. So that's really the main distinguishing characteristic, at least with regard to these tetrad structures. Okay, so once diakinesis is complete, again, what's going to happen is you're going to transition now into metaphase 1. So here's metaphase 1 of meiosis. Again, what you can see now is the two tetrads that I've been describing are now properly aligned along the central axis of the fully formed spindle apparatus. So once that occurs, you can then, or the cell can prepare to transition into anaphase. And again, remember what happens during an anaphase is that you get segregation of the genetic information, the chromosomes that have been aligned at the metaphase plate. So that's what's happening here. That's what's happening in anaphase 1. And what I want to point out is that what's happening here is a fundamentally distinct type of segregation relative to the segregation that takes place during anaphase of mitosis. So during anaphase of mitosis, what happened is that sister chromatids segregated to opposite poles of the spindle. But that's not what's happening here. That's possible because every chromosome aligns independently of every other chromosome at that mitotic metaphase plate. But here you have these tetrad structures that have aligned. So each tetrad structure actually already consists of two centromeres. So one centromere within the tetrad structure is pulled to one pole of the spindle, and the other centromere is pulled to the opposite pole of the spindle. So this is what we call a reductional segregation. I'll get back to that as to why we call it that in a second. But the idea is that during anaphase 1 of meiosis, there's a fundamentally different type of segregation that is taking place relative to a mitotic anaphase. In this case, for a reductional segregation, what you have are replicated homologous chromosomes segregating to opposite poles of the spindle. That's a reductional segregation. So once that segregation is complete and the chromosomes aggregate around the two poles of the spindle, you transition into telophase. And in telophase, you reassemble a nuclear envelope around the two aggregates of chromosomes. The spindle apparatus is destroyed or broken down, and the chromosomes within the nucleus will then decondense back into a chromatin-like state. And then again, during the telophase, you also have another event taking place that is segregating cytoplasmic contents of the cell as well. And so again, the process of cytokinesis. So the end result is you produce from this first meiotic cell division is two daughter cells. Okay, and the process is then complete. So these two daughter cells will then enter into the second meiotic cell division, meiosis 2, and again proceed through a prophase, a metaphase, an anaphase, and a telophase. Okay, before I describe those events, I want to get back to this, what I called earlier a reductional segregation here. So what does it mean to be reductional? So it's the idea is that, again, remember in this meiotic cell division that we're describing, we started with a diploid cell, a 2N of 4. Well, these two daughter cells that result at the completion of meiosis 1 are now haploid. They no longer contain 4 chromosomes, they now only contain 2 chromosomes. And that's because of the reductional segregation that has taken place during this anaphase. So again, when you have chromatids segregating to opposite poles of a spindle, that's an equational segregation. You have identical genetic information segregating to opposite poles of the spindle. But with a reductional segregation that's going to occur during the first meiotic anaphase is that you have replicated homologous chromosomes segregating to the 2 opposite poles of the spindle. So an original cell that had 4 chromosomes now produced 2 daughter cells that only contain 2 chromosomes. So that's one of the fundamental goals. Again, remember, meiosis is producing these reproductive cells, these gametes, these haploid cells. And how are they able to produce these haploid cells? It's because of this reductional segregation. And why does the reductional segregation take place? Because of the pairing of homologous chromosomes that took place during early prophase prophase 1 of meiosis. So that pairing allows for this unique type of segregation that produces the haploid cells. Okay, the other point about the formation of those, or the pairing of those homologous chromosomes, is it leads to the formation of the tetrad. And what happens, what did I describe earlier, that takes place within that tetrad is that you have genetic recombination events. So again, the formation, the pairing of homologous chromosomes lead to genetic recombination events that allow this process of meiosis to produce as final products genetically diverse cells, genetically diverse haploid cells, because of the pairing of those homologous chromosomes very early on in prophase 1 of meiosis. Okay, so in any event, once these 2 haploid cells are produced, they transition into the second meiosis, second meiotic cell division. So there's a prophase, again, the same characteristics that I've described previously for prophase stage of a cell division. You then transition into a metaphase, same characteristics as I've described previously. And then you transition into an anaphase, so anaphase 2 of meiosis. So what's happening during this anaphase 2? What type of segregation is taking place? It's the exact same type of segregation that occurs during a mitotic anaphase. You see that there are chromatids that are segregating to opposite poles of the spindle. So it's an equational segregation, just like a mitotic anaphase. So what that means is that the end result of this process, this is equational. Remember, we started with meiosis 2, we started with haploid cells. That means the final products of meiosis 2 are going to be haploid as well. It's equational. Okay, so here's the anaphase, and then you transition into telophase again, the same characteristics as I've described previously. You have cytokinesis taking place at the same time as the telophase events, and then the final meiotic products are produced. There's a total of 4 haploid cells, and all 4 of those haploid cells are genetically distinct from each other. And the mechanism that allows for that, again, as I said, are the genetic recombination events that take place within the tetrad. So again, the goals of meiosis have been achieved through this unique form of segregation, a meiotic type segregation. Okay, so I just want to point out is that what I've described to you is the process of meiosis, very generically, and I just want to point out, for certain types of organisms, and again, we've been using humans as the example, some unique events that take place, during human meiotic events. And so again, as I said, the starting material within the testes of the males are spermatogonial cells. These are 2N cells that have the ability to commit to a meiotic event. They proceed again through a first meiotic cell division, or that first division of meiosis, to produce, again, two daughter cells, which are now haploid and genetically distinct from each other because of the recombination events. Then you have the second meiosis, second meiotic cell division, that produces the final meiotic products, these four haploid cells that are still immature. So these four cells have to go through a process of maturation or differentiation before they produce the final functional spermatozoa or sperm cells that have the capability of participating in a reproductive event. What I want to point out here is the idea of this process of cytokinesis. Is through this process, meiosis 1, meiosis 2, and in particular during this differentiation process, most, if not all, of the cytoplasm found within the original cell is lost. So the final mature spermatozoa cells are essentially a nucleus with very little, if any, cytoplasm associated with them. So that's a little bit different than, again, sort of the generic process of meiosis that I've described previously. On the female side, again, it's different from, the generic pattern that I've described for cytokinesis. So again, starting with a 2N cell, an oogonial cell, that's a cell within the ovary that can commit to a meiotic event, proceeds through meiosis 1, telophase, and cytokinesis. You end up with two daughter cells, which are now haploid and genetically diverse. However, what happens during cytokinesis is that there's an asymmetric distribution of the cytoplasm into those two daughter cells, so that one cell receives the bulk of the cytoplasm and the other one receives very little. And so that the one that receives very little is what we call a polar body, or the first polar body, and that cell is generally incapable of further division. So it arrests at that stage. This other cell that receives the bulk of the cytoplasm can proceed through the second meiotic division, and that's what happens. So two daughter cells result. And again, there's another asymmetric distribution of the cytoplasm during cytokinesis of telophase. So that again, only one of these two daughter cells remains functional. In other words, can differentiate into a mature ovum or a mature cell that can be fertilized by a sperm. The other cell that receives the least amount of cytoplasm is a polar body, and again is arrested at that stage, and generally will die and not mature into a functional reproductive cell or gamete. Okay, so I've tried to describe meiosis again and describe some of the specific mechanisms and events that allow for the unique goals of meiosis to be achieved. So again, the goals of meiosis are to produce genetically diverse haploid cells from an original diploid cell. Okay, so we saw the genetic diversity being generated through the mechanism of recombination. Again, the recombination that takes place within the tetrad. There's actually a second mechanism that is operating during meiosis that further increases genetic diversity among the final products of meiosis. And so that's what's being described in this diagram here. So for our example, again, we're going to have a starting point, a 2N cell of 4. So a diploid cell consisting of two homologous sets of chromosomes. So this cell is now going to enter into meiosis. So what happens, you transition through the S phase. Chromosomes, these 4 chromosomes will duplicate themselves. And then you'll transition into prophase 1. You'll form the tetrads and so on. So here are the tetrads. So what I want to point out in these tetrad structures is that, again, the 2 different chromosomes among each member of a homologous pair are being distinguished. You see they're represented as either a solid black chromosome or a sort of a gray or a stippled chromosome. Okay, so again, that distinguishes between maternal and paternal members of the homologous pair of chromosomes. And again, remember that that's an important distinction because of the allelic variation that can occur between the two members of a homologous pair of chromosomes. So you've got, again, one maternally derived chromosome for this set. The maternal are the stippled chromosomes or the gray chromosomes here and here. And then you have the paternally derived chromosome, or the one derived from the father, that is black here and here. So these two tetrads, again, will proceed through meiosis one and enter into metaphase. And so here's these two tetrads now aligned at the metaphase plate. But actually what you see is there's two different depictions of that same metaphase one. So what that means is that these two tetrads that form during prophase one, they can align relative to each other at that first metaphase of meiosis in two different ways. That's what's being depicted in this portion of the diagram. So how are they different? So look at the bottom depiction first, this metaphase. You can see the two paternally derived chromosomes, the black chromosomes, are aligned to the left, and the two gray or stippled chromosomes, the maternally derived chromosomes, are to the right. So that's one configuration as to how those two tetrads can align relative to each other at that first metaphase. But there's another equally probable alignment, and that's what's shown here. So here, the paternally derived chromosome of this large chromosome is aligned to the left, but the paternally derived chromosome of this smaller chromosome is aligned to the right now. And then the same thing on opposite sides of alignment of the metaphase plate for the two maternally derived chromosomes. So that's different from what's being depicted down here. So each of these arrangements, each of these alignments for the two tetrads are equally probable. So what that will lead to, the idea that there's random alignment of tetrads at that first metaphase plate, what that leads to is what we call independent assortment. And that's a concept that we'll get back to when we talk about Mendelian genetics in an upcoming lecture. So I'm describing independent assortment at a chromosomal level now, and later on we'll talk about it at a more genetic level based on the observations of Gregor Mendel. So if you then take these two alignments and you follow them through completion to the end of meiosis one, these would be the two daughter cells that would result from this alignment, and these would be the two daughter cells that would result from this alignment. So again, you can see all four of those cells now are haploid. We're ignoring recombination at this stage, but so all four of these cells are haploid. So then if you follow each of these four cells, 1, 2, 3, and 4, through the second meiotic cell division, these are the final gametes, the final haploid meiotic products that are going to be produced from each of these four cells. And if you look at them, and look at them closely and carefully, all four of them are genetically distinct from each other. So here you have two gray chromosomes. Here you have two black chromosomes. Here you have the larger gray chromosome and the smaller black. And here you have the larger black chromosome and the smaller gray. So all four of those cells, those final meiotic products, are genetically distinct from each other. And they're genetically distinct from each other because of the random way that these tetrads align relative to each other at that first metaphase plate. So genetic diversity is being created through independent assortment. So the independent assortment takes place through the anaphase, anaphase one. Okay, so this mechanism is really important. I mean, it's as equally important as the recombination, genetic recombination that I described earlier taking place within the tetrad, important in terms of generating genetic diversity. So you can see in this case with a 2n of 4, two pairs of tetrads that are, or two pairs of homologous chromosomes that are present in the genome, simply by independent assortment, this particular mechanism, you can create 4 genetically diverse meiotic products. Here's sort of an extension of that. We'll skip that. What if you had a 2n of 6? So 3 pairs of homologous chromosomes in the genome. And again, distinguishing maternal and paternal, with different colored uh, chromosomes. So each homologous pair consists of 1 purple chromosome and 1 green chromosome. So maternally derived, paternally derived, and so on. So if you had a 2n of 6, 3 pairs of homologous chromosomes in the genome, what they're showing you here is simply based on independent assortment, you can produce 8 genetically distinct meiotic products. Because there's 4 different ways that these 3 homologous pairs of chromosomes can align relative to each other at that first metaphase plate of meiosis. And you should be able to figure that out. In other words, kind of walk through this particular genome, the 2n of 6, through meiosis, and see for yourself how you would generate these 8 genetically diverse final products of meiosis based solely on independent assortment. And if you can't do that, you should ask about it. Either ask me or ask the TAs during a recitation section. So the sort of the general way of looking at this and an indication of the significance of independent assortment as a mechanism for producing genetic diversity during a meiotic event is summarized in this formula down here, where 2 raised to the nth power, where n is equal to the number of homologous gene pairs within an organism's genome. So again, with the previous example, a 2n of 4, there were 2 homologous sets of chromosomes within that genome. So 2 raised to the 2nd power, you can produce a total of 4 genetically distinct gametes based solely on independent assortment. In this example, a 2n of 6, 3 pairs of homologous chromosomes, 2 raised to the 3rd power is 8. So to extend that to the human situation, again, remember our diploid number is 46. So we have 23 pairs of homologous chromosomes in our genome. So 2 raised to the 23rd power is a number that's greater than 8 million. So simply based on independent assortment, all humans have the ability to create over 8 million genetically distinct gametes, simply based on independent assortment. But remember, there's a second mechanism for creating genetic diversity during meiosis. It's genetic recombination. So that means there's essentially limitless genetic diversity that is created through these 2 mechanisms during meiotic events. Okay, so again, what does that allow for? It allows for the maintenance within populations of organisms across generations, maintaining maximum levels of genetic diversity, assuming random matings occurring within those populations. Okay, so this is just a summary slide of all of the points we've tried to make in comparing and contrasting mitosis and meiosis. And I just want to point out a few general distinctions. So again, in all instances, we're starting with a diploid cell. So again, proceeding through mitosis, what happens is that during the metaphase is every chromosome, whether they be homologous or not, align independently of every other chromosome at that metaphase plate. That leads to an equational type of segregation during the anaphase, so that you can then see that equational segregation, sister chromatids segregating to the two opposite poles of the spindle, allow for the maintenance of constancy of genetic information through the cell division. So again, here we started with a diploid cell. These two daughter cells that result from the completion of mitosis, again, are diploid. So what's the fundamental difference in meiosis? So that you can achieve the end result of genetically diverse haploid cells, these specialized cells that are used for reproduction. Well, you form that tetrad very early on in the process during prophase 1, again, the very early stages, zygonema. Those tetrads allow for genetic recombination to take place during prophase. They then allow for alignment of the tetrads at the metaphase plate, the first metaphase plate. So again, you have paired replicated homologous chromosomes that are aligning together. That leads to the reductional segregation that takes place during that first meiotic anaphase. And the end result of that is that you produce two daughter cells that are now haploid, as well as genetically diverse because of independent assortment and because of genetic recombination. So the requirement for the second meiotic cell division, this equational cell division, essentially equivalent to a mitotic event, is that every one of these chromosomes are still in a duplicated form. So you have to segregate the chromatids away from each other to produce the final meiotic products. So meiosis 2 is essentially equivalent to a mitotic cell division. The anaphase exhibits equational segregation. So you start with a haploid cell, you end up with haploid cells. Here's another table, a slightly different way of summarizing the differences between mitosis and meiosis. Really, the only thing I want to point out here is the idea that for mitosis, because there's not a requirement for pairing of chromosomes at any stage of the process, is that a haploid cell can undergo a mitotic event. So you can start with a haploid cell and it can divide mitotically. But typically you start with a diploid cell. Okay, for meiosis to work, you have to at least minimally have a diploid cell. A haploid cell would be unable to undergo meiosis because there wouldn't be two copies of any given chromosome. You wouldn't have homologous pairs, and therefore you can't form a tetrad. You can't get reductional segregation, et cetera, et cetera. You can't get recombination and independent assortment, all of that. So minimally for meiosis, there's a requirement, there's the need that the cell at least be diploid. Okay, this is one other characteristic of mitosis and meiosis that I want to describe. So this is the idea that, so what I've been doing in these reviews of the two processes, mitosis and meiosis, I've been describing what's happening through the cell division with regard to looking at the chromosomes. Okay, well, there's another way as a geneticist you can describe the processes. You can describe them in terms of changes in terms of the actual genetic content of the cell. In other words, the DNA content. And when you look at them from these two different perspectives, the changes that occur are a little bit different. So what do I mean by that? So again, when you're describing chromosome changes through cell divisions, you describe ploidy levels. So, we've previously defined haploidy and, a haploid condition, a diploid condition, and so on. So you count chromosome numbers, based on these ploidy levels. So again, remember, for fertilization to occur, to begin the life of an individual, you have to start with two haploid gametes. So a 1N chromosome constitution, those two gametes fuse in a fertilization event, and immediately you double the chromosome number So you generate a diploid state within that zygote. So now, again, what happens with that zygote, again, in a multicellular organism, is it begins to divide mitotically to create large numbers of cells that will assemble, the body of the individual. So as you proceed through these mitotic events, you pass through the S phase, you enter mitosis. The A represents an anaphase. You go to completion. The cells divide, the daughter cells divide again. They pass through an S phase, transition through an anaphase, and so on. So what's happening to the chromosome number in those cells? Well, it's remaining the same. It's unchanging. So in other words, if here you have a diploid cell through the S phase and through the mitotic cell division, chromosome number remains unchanged. So this is the idea of maintaining genetic constancy through a mitotic cell division. Okay, so chromosome number remains unchanged. Again, if you started with a haploid cell, the daughter cells of a mitotic cell division would be haploid, so you maintain constancy. Okay, so what's happening during a meiotic event? So it's different. Again, remember, during a meiotic event, you start with a diploid cell, but the end result are haploid cells. So when do you actually generate those haploid cells? Well, I've described it previously, is that it occurs during the anaphase of the first meiosis. So here, now you have a cell that's entering into a meiotic event, passes through the S phase. It doesn't change chromosome number. You transition through anaphase 1. Because of the reductional segregation, the daughter cells that result are now haploid. And then through the second meiotic cell division, again, remember it's equational, it's essentially equivalent to a mitotic cell division, is that the chromosome number remains the same. It remains at a haploid level. So this is describing the changes in chromosome number through mitosis and meiosis. So you can describe the same events, but instead look at changes at the actual, with the actual DNA or genetic content of the cell. So how do you measure or how do you quantify DNA content? Well, they use this C value. So a haploid cell would consist of a 1C amount of DNA. A diploid cell would be 2C. If you had 3 copies of every chromosome within the genome, it'd be a 3C level, a 4C level, and so on. So this is measuring DNA content. So a haploid cell has a 1C. Again, remember, there's one DNA molecule that runs from end to end through any given chromosome. Okay, so those 2 haploid cells will then, those gametes will fuse in a fertilization event and immediately double the DNA content of the cell to a 2C level. Okay, so a diploid cell would be at a 2C level. So then what happens is then through mitosis is that you're going to enter into an S phase. And again, remember, during the S phase, the DNA molecule duplicates itself, which are seen at the chromosomal level as chromatids. So through the S phase of the cell cycle, you've doubled the DNA content. So if you start with a diploid cell, you've now doubled the DNA content to a 4C level. Then through mitosis, what happens is the nature of segregation that takes place during that mitotic anaphase is that the DNA content of the daughter cells is cut back to a 2C level. And then these changes in DNA content again continue through subsequent mitotic cell divisions. So the S phase doubles the DNA content, and the DNA content is reduced through the anaphase of mitosis. So what about DNA and that continues through every mitotic cell division. So what happens during a meiotic event? So it's the same idea as you start with a diploid cell with a 2C DNA content. That DNA content doubles during the S phase of the cell cycle. You then enter into that first meiotic cell division. And during that first anaphase, what happens through the reductional cell division is that you cut the DNA content in half. So you have two replicated copies of each homologous chromosome set migrating to opposite poles of the spindle. So the DNA content is cut in half. It goes from the 4C level to the 2C level. And then through the second meiotic cell division, the second anaphase, that's when you have chromatids segregating to opposite poles of the spindle. And therefore, you're reducing the DNA content again by half from the 2C level down to the 1C level that you would expect to see in the final meiotic product, which is a haploid cell. Okay, so this is, again, just a diagram. I'm not going to go into all of the details of describing, but this, again, just summarizes what I've just described. The changes that occur in terms of chromosome number and DNA content through mitotic cell divisions and meiotic cell divisions. Okay, so with that, I'm going to wrap up this discussion. Well, we're going to, with this background, you know, the reason I spent so much time discussing these processes is that, in particular, with meiosis, what we know nowadays is that the observations of Gregor Mendel, who knew nothing about chromosomes, his observations in terms of inheritance patterns for traits in the pea plant are explainable based on these segregation patterns for chromosomes that take place through a meiotic event. So we're going to talk about Mendel's work in general, how he interpreted those results, and the conclusions that he was able to draw from those observations. And then we're going to begin to relate those observations to a more modern day understanding of genetics, knowing that his unit factors of heredity, what we now call genes, are localized on chromosomes, and therefore their inheritance patterns can be explained based on chromosome segregation patterns that take place during meiosis.